Retired Professor Dr. Walter Bradley, who has more awards and honors than most any other professor I've seen in quite a while, has an interesting paper entitled "Is There Scientific Evidence for the Existence of God?" And I'd like to focus in on one particular spot in his paper where he goes over the fine-tuning of light and the fine-tuning of biological life for each other. He has 4 charts, and he states the visible portion of the electromagnetic spectrum is the most intense radiation from the sun. Figure 1 has the greatest biological utility, figure 2, and passes through the atmosphere of the Earth, figure 3, and through water, figure 4, with almost no absorption. It is uniquely this same wavelength of radiation that is ideal to foster the chemistry of life. This is either a truly amazing series of coincidences or else the result of careful design. At the 36-minute mark of this video, Dr. Walter Bradley goes a bit deeper into this.
As another example, we need to balance or match the energy, as I mentioned before, in light to the bonding energy in chemicals. The electronic energy levels and the chemical bonding energy levels that come from those are determined from quantum mechanics, and they depend on the mass of the electron, the unit charge, and Planck's constant, with an equation that looks something like this. Okay, bonding energy between these different atoms. The wavelength of light, if it's going to be tailored to the chemical bonding energies, would require a spectrum which has its highest intensity in what we call the visible spectrum. This visible spectrum, which in fact we have, requires satisfaction of the following algebraic equation. Now, don't let the equation intimidate you here. It's nothing more than algebra. This is an inequality sign. This is approximately equal to sign. The term on the left must be greater than, but of the same order of magnitude as the term on the right. And the constants in this equation are, in fact, Planck's constant, speed of light, gravity force constant, mass of the electron, mass of the proton, and the unit charge. Several of these were required in the previous requirement for specifying chemical bonding energy. If I take all of these values from the chart I showed you previously and substitute them into this equation, lo and behold, I get a result that looks like this. The left term is, in fact, bigger than the right term, but they are both of the same order of magnitude. And what this says is that the selection of these constants, in fact, is in exactly the right proportion to each other, as required by this equation, to allow the energy from the sun to be exactly right, to be interactive with chemical bonding in the periodic chart that we have. There are a couple of other interesting things that come out of this that are not specified in that preceding equation. If I'm going to have visible light as the primary source of energy to drive the necessary chemical reactions in living systems, then it's important that this radiation not be highly absorbed by the atmosphere or by water, because most of the important biochemistry occurs in a water or aqueous solution. If you look at the absorption as a function of wavelength of light, what we find is that almost all wavelengths of light are highly absorbed by water and by the atmosphere. Only in this very narrow range of visible to infrared is there very, very little absorption of that very same wavelength of radiation that's necessary to drive our chemical reactions. And the same thing, surprisingly enough, with our atmosphere. So it turns out not only is the energy of light, the primary wavelength of light from the sun, exactly what's needed for chemical reactions, it also happens to be very low in its absorption with water or with the atmosphere. If the light we needed or the radiation we needed was someplace either here or here, it would actually help chemical reactions, but it would never get to the chemicals because it would be highly absorbed.
This clip from Privileged Planet, Chapter 9 of 12, goes a bit deeper.
Our appreciation of the Earth's atmosphere has increased significantly during the last 40 years, as exploratory spacecraft have probed the solar system. These missions have confirmed that within the Sun's family of more than 70 planets and moons, the Earth is one of 7 bodies enveloped by a thick canopy of gas. Yet among these 7, only the Earth's atmosphere can sustain complex life. And only the Earth's atmosphere is transparent.
It's an atmosphere that's made up of mostly oxygen and nitrogen with very little carbon dioxide and very little other carbon compounds or atoms in the atmosphere that gives you a transparent atmosphere. If we had too much carbon in the atmosphere, we get hazes, organic hazes in the atmosphere, like you see on the large moon Titan, for example.
The dense shroud of gas that blankets Saturn's largest moon resembles the atmospheres surrounding Neptune, Uranus, Saturn, Jupiter, and the greenhouse cauldron of Venus. None of these alien worlds know the stars, or even offers a clear view of the sun.
Now, of course, if you were suddenly transported to Titan or Venus or to one of the outlying gas giant planets, the lack of a clear view of the universe wouldn't be much of an issue because you'd be dead. But that's precisely the point. If we're right, if the conditions for habitability and scientific discovery appear in the same places, then you're going to get conditions like you do on Earth, an atmosphere that sustains complex life like ourselves and also enables scientific discovery of the universe around us.
The virtues of such an atmosphere are continually tested. As the Earth moves through space, it is bombarded by radiation from throughout the universe. This radiation is emitted by the Sun and other celestial objects, including supernovas and distant galaxies. It reaches our planet in wavelengths described as gamma, X-ray, ultraviolet, visible, infrared, microwave, and radio. Together, they comprise the electromagnetic spectrum. Almost all of these wavelengths are invisible to the eye and either lethal or useless to organic life. Yet within this spectrum of frequencies, a thin sliver of radiation proves essential to plants, animals, and human beings.
In other words, there's really just a very narrow part of the electromagnetic spectrum that's going to be useful for living processes like photosynthesis. It's not as if life could have evolved to use gamma radiation or X-ray radiation or something like that. There's really just a narrow part of the spectrum that would be useful to life processes. Well, as it turns out, that's also the same narrow part of the spectrum that is the most informative about the various structures that we discover in the universe around us.
These specific frequencies that enable plants to manufacture food and astronomers to observe the cosmos represent less than 1/1,000,000,000th of a trillionth of the universe's range of natural electromagnetic emissions. Fortunately, it is the type of light our sun produces in abundance, and that most easily penetrates the filtering shield of our atmosphere to reach the surface of the Earth.
It's a remarkable coincidence that the kind of atmosphere that's needed for complex life like ourselves does not preclude that life from observing the distant universe. It's a surprise. It's something that you wouldn't expect just chance to produce. Why would the universe be such that those places that are most habitable also offer the best opportunity for scientific discovery?
Here's an article entitled "The Just Right Relationship of the Light Spectrum and Photosynthesis," which gives a good overview of the preceding 2 video clips. You can read it at your leisure. I just want to draw attention to the last line in the Encyclopedia Britannica remarks, "Considering the importance of visible sunlight for all aspects of terrestrial life, one cannot help being awed by the dramatically narrow window in the atmospheric absorption and in the absorption spectrum of water. Moreover, besides the fine-tuning of sunlight and atmosphere and water and biological molecules, we also find that in this article, quantum mechanics explains efficiency of photosynthesis. The authors state, we found that the properties of some of the chromophore vibrations that assist energy transfer during photosynthesis can never be described with classical laws, and moreover, this non-classical behavior enhances the efficiency of energy transfer. It should be noted that quantum coherence is very similar to quantum entanglement in that it requires a non-local, beyond space and time, cause in order to explain its effect. In this article, it states, whereas the EPR, non-local entanglement connections, are fragile, some forms of non-local coherence are robust. As to how efficient this non-local, beyond space and time quantum process is, this article states certain biological systems living in low light environments have unique protein structures for photosynthesis that use quantum dynamics to convert 100% of absorbed light into electrical charge. These biological systems can direct a quantum process, in this case energy transport, in astoundingly subtle and controlled ways, showing remarkable resistance to aggressive random background noise of biology in extreme environments. It should be noted that photosynthesis absolutely blows away anything man has built in terms of efficiency. This article states the very best solar panels ever tested in a lab, not the ones actually available for sale and installation on your house, were able to convert about 34% of the light that hit them into electricity. This article states the photosynthetic system of plants is nature's most elaborate nanoscale biological machine. It converts light energy at unrivaled efficiency. Photosystem 2 is the engine of life. It performs one of the most energetically demanding reactions known to mankind, splitting water, with remarkable ease and efficiency. The 4-photon reaction has very stringent requirements. The following video gives a short overview of nature's most elaborate nanoscale biological machine, the photosynthetic system of plants.
The following complexes are found in the photosynthesis electron transport chain: Photosystem 2, cytochrome B6F, Photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP, and the electrons to form NADPH, and ADP and PI, which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem II complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem II, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. One photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem II are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem I complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
In the following video, Dr. Krish Ashcraft gives a short overview of ATP synthase and the citric acid cycle.
Now, movement, just like moving muscles, moving our bodies, cellular movement as well requires energy, and that energy is provided by this molecule that's called ATP. Okay? So this is the molecule that carries energy. It's an energy carrier for anything that an organism needs to do. Okay? And it supplies that energy to the molecular machines, which are themselves proteins in almost all cases, by cleaving off this portion of this molecule. All bonds between atoms, this is a big molecule, right, and all these letters represent different atoms like oxygen and stuff, and so it cuts off this last little phosphate group and then transfers that energy to the protein when the protein needs to do something, okay? So this is the energy carrier for the cell, ATP. You may also recognize that this is one of the 4 components that make up DNA. ATP serves as an energy carrier and one of the 4 components of DNA, okay? But your body actually makes and uses half your body weight each day in ATPs. Did you hear that? Half your body weight each day in ATPs is how many ATPs you go through. And cyanide, you know, the poison cyanide, it blocks ATP synthesis, and in doing so will kill you in 30 seconds. That shows you how badly you need ATP. ATP is required for all things. Glucose, which is the blood sugar, you know, blood sugar levels, you know, that's glucose. Glucose is eventually converted into ATPs. So glucose is an energy carrier, like a $100 bill, but your cell only needs quarters to do things. So it converts glucose molecule into essentially 36 ATPs through a process we call cellular respiration. Now I'm going to have to hit you with some biochemistry, okay? A little bit of biochemistry, but it will be relatively painless. Relatively painless. Okay? This is the molecular machine that makes ATPs right here, called ATP synthase. The smallest molecular machine we know of. The person who discovered this was awarded the Nobel Prize in 1997. Just so, you know, things were still pretty recently, we've got to think. What it does, this thing is composed of like 500 separate subunits, and basically this head spins at 10,000 revolutions per minute. So this head on this thing is spinning right here, and it makes 3 ATPs with every revolution. So basically, this is just a crude little animation which shows, makes an ATP, 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 ATP. As this thing is spinning, it's cranking out ATPs. Okay? But this ATP synthase machine doesn't work by itself. Okay? Now, I don't expect you to understand everything that's going on here. I just want to impress upon you the complexity of metabolic pathways that are responsible for making things like all-important ATPs. This ATP synthase doesn't work by itself. It's part of a long process, a metabolic process that starts with glycolysis and then citric acid cycle and then electron transport chain. There's a whole bunch of reactions that go on here. Now, this is just part of it. Basically, glucose is broken down into this one molecule called pyruvate, and then this molecule is basically converted into this molecule, that's converted into this molecule, that's converted into that molecule, then to that molecule, then to that molecule, then that. And that's what a metabolic pathway is like. But each of these conversions to convert that molecule into this one, citrate into isocitrate, requires a protein. An enzyme is a protein that performs chemical reactions. Okay? So there's an enzyme here that converts this one. There's another enzyme here, isocitrate dehydrogenase, does this one. Alpha ketoglutarate dehydrogenase does this one. There's another protein does this one, another protein does this one, another protein does this one. But this thing doesn't work unless you have all of these proteins. And there's no purpose for it except to ultimately make ATP. Now, what the evolutionists have to explain is how all of these proteins came into existence. A metabolic pathway like this, these are not uncommon, but it really doesn't work. There's no purpose for this except to make this. Now, in some cases, some of these molecules do have other purposes, but metabolic pathways are generally like this, that there's no purpose of this molecule except to make this one. There's no purpose for this enzyme except to convert this to this. And unless you have the entire thing present, it's just not going to function. But what makes it even more remarkable, like I say, you have glycolysis, citric acid cycle, electron transport chain, which ultimately drives this little motor to make ATP. All of this requires ATP. So you have to have ATP to drive glycolysis and then to feed this pyruvate into this, what then ultimately turns this. The whole thing requires ATP. Extremely, extremely complex. So,
and this article states this motor, ATP, is always near perfect under different ATP hydrolysis energy conditions. This near perfect level of efficiency is maintained out throughout a broad range of operation conditions. And that's the end of the video. Thanks for watching.